Let's start to look at a neuron. And once we learn some basic parts to a neuron, we can better understand how a neuron is going to function, as well as how neurons are going to transmit information throughout the body in the form of action potentials. So neurons are excitable cells that transmit electrical signals. You'll recall that muscle cells are also excitable cells. And what this means is they can respond to a stimulus and convert that stimulus into what we would call an action potential. An action potential is a change in the electrical activity on the inside versus the outside of the plasma membrane. But the whole idea of an action potential we're going to get into once we review some of the anatomy of neurons. Neurons happen to be a very long-lived cell. They divide at an early age and during fetal development, but then they stop dividing. So we often call them amitotic or postmitotic. That means they have stopped dividing. Mitotic referring to mitosis. So all the neurons you have now. And if we lose neurons, they're gone forever. So we have to protect our neurons. We have to do things to keep our neurons healthy, like study anatomy and physiology. That keeps the neurons of your brain very active. It makes them get better at what they're doing. Neurons are very highly metabolic. They have a high metabolic rate. What that means is they're going to require a lot of glucose. They're going to require a lot of oxygen. So your brain gets a lot of your blood flow by percentage, even though, if you remember, the nervous system makes up very little of you by mass. It gets a lot of your energy supplies. So it requires a continuous supply of oxygen and glucose, and therefore, if you have a compromised cardiovascular system whose job is to deliver and supply oxygen and glucose, typically your brain is the first one to feel the effects of low oxygen and low blood sugar. We're going to be using this picture. This is your textbook's drawing of a neuron. And we're going to use this picture to identify some of the parts of a neuron. I'm going to go to the next slide, but you'll want to refer back to this drawing as I go over the structures of a typical neuron. Okay, so there's in essence three main components to neurons, and there's some variation between these components, and there are even more accessory parts as well. But the three main components include the cell body, the dendrites, and then the axon. The cell body is also known as the soma. Soma means body. This is where you're going to find the nucleus. So neurons have a single nucleus found inside its cell body, and a highly developed Golgi system. This Golgi system is going to be playing a big role in production of the neurotransmitter that's released by the neuron. Typically, the cell bodies are located either in the CNS or very close to the CNS in a structure called the dorsal root ganglion, or ganglia. We'll be talking more about this dorsal root ganglia, especially when we get to the spinal cord. Couple terms to be familiar with. Quite often you'll find neurons tend to be packed together, and quite often wherever the cell bodies are packed together, that area is typically is gray matter, and we'll talk more about what gray matter means. But when you have a lot of cell bodies packed together in the CNS, like in your brain, we often refer to those areas as nuclei or basal nuclei. But that's just because there are these areas that have a lot of cell bodies and therefore no myelin, and that area is gray matter. But we'll be talking more about that when we get to the brain. A lot of times when you have a lot of cell bodies packed together in the periphery, we call that a ganglion or ganglia. The word ganglia actually means swelling. So where the nerve has a lot of cell bodies, it'll look like it's swollen because the cell bodies make up a large majority of the size of a cell because of that large nucleus. So if you see these two words, try to recall that what that just means is you've got a lot of cell bodies in the same location. Dendrites. Dendrites are typically short little processes that are sticking off of the cell body. So if you look at a picture of a neuron, you'll typically see off of that cell body these small little structures projecting from them. And the most important thing to remember about the dendrites is that's where the neuron will get stimulated. So that's what its receptive portion. That's the portion of the neuron that's either getting stimulated by some sort of mechanical stimulus, maybe a touch receptor is on the dendrite, or some sort of chemical stimulus. So maybe that's where it has receptors for neurotransmitters. And when a neuron is getting stimulated, that's going to cause what we call graded potentials. And we'll be talking much more about what these are. But a graded potential is a small change to the electrical activity at the dendrites. And these small changes are then going to either add up to an action potential or not. But again, that's a topic for another day. But keep in mind that the dendrites are going to have receptors. And those receptors are how that neuron is getting stimulated. The different types of receptors are largely based on what kind of channels they are. They could be a ligand-gated channel, and the word ligand or ligand refers to a chemical, usually a neurotransmitter, or it could also be some other maybe inflammatory chemical. But when you see the word ligand or ligand, just think of a chemical as opposed to, say, a mechanical stimulus. So that's how neurons get stimulated. It's either some chemical that's stimulating them, or it's a mechanical stimulus like pressure or touch or vibrations. But those channels are going to open up in response to the stimulus, are going to allow ions to enter or exit the neuron, and that's going to change the electrical activity of the neuron. All physiology that we'll be getting to momentarily. The axon is also a process that extends from the cell body, but it's typically very long because the axon is what's going to have to send the nerve impulse to the effector. The effector is whatever the neuron is then controlling. The effector could be another neuron. The effector could be a muscle. The effector could be a gland. But whatever that neuron is controlling, it has to have an axon that extends to that organ or that structure. So the axon carries the nerve impulse or action potential away from the cell body to the end bulb. And we've already seen what an end bulb is. That's the terminal end of the axon that contains synaptic vesicles, and we already know what's in the synaptic vesicles. It's whatever neurotransmitter that neuron releases. Along the axon, there are channels that are going to be extremely important for us to understand how an action potential works. Those channels are voltage-gated channels. So what opens these channels is a change in the voltage along the axon. There are also a lot of sodium-potassium pumps, which we're going to learn their role is to maintain proper amounts of sodium and potassium either on the inside or on the outside of the cell. Specifically, these sodium-potassium pumps move sodium outside of the cell and move potassium inside of the cell. There are some parts to an axon. For example, where the axon comes off of the cell body, it's usually a cone-shaped area known as the axon hillock. Then as that axon comes from off of that axon hillock, there's an area called the initial segment. That's basically the initial segment of the axon. So it's the first part of that axon. And usually that's where the nerve impulse begins. The nerve impulse begins, or the action potential begins, on the initial segment. Whenever you have a lot of axons bundled together in the CNS, we call it a track. When it's in the PNS, we call it a nerve. I know we've heard of nerves before, but a nerve is just a bundle of many axons. So there's a lot of neurons that are found in a single nerve. Tracks are kind of like nerves in your brain and spinal cord. They're just a bundle of neurons all going to a similar or same location. So these are all parts of that typical neuron. If we go backwards very quickly, I also want to point out, again on the drawing, here's the cell body. These are the dendrites. So they're going to get this information, or they're going to be stimulated. Here is that cone-shaped portion called the axon hillock. Then here about they say is the initial segment. Notice the arrows represent the electrical activity that's going to be moving down the axon. Notice this must be a peripheral nerve because it's showing Schwann cells surrounding portions of the axon and producing myelin sheaths. And we're going to learn that what that's going to do is allow the action potentials to jump. The action potentials actually jump over the Schwann cells. And that's how this can speed up nerve transmission. And then right here at the end are the endobulbs that are going to be releasing the neurotransmitter, whatever neurotransmitter this neuron uses. So those are the basic components of a neuron. Neurons also come in different structural categories. We'll go quickly through the structural categories, but it's all based on how many things, how many processes are seen projecting from the cell bodies. If there's many processes, like this one here, it's known as a multipolar neuron. Those are typically the motor neurons and interneurons, which is a type of neuron in between two neurons found in the CNS. If it's got two things, two processes, usually one's an axon and the other one's a dendrite, it's called a bipolar neuron. Bi meaning two, so two processes extending from the cell body. Those are very common in your special senses, like your olfactory epithelium for smell and the retina of the eye. And then finally, there are unipolar neurons, which have one process sticking off the cell body, but it usually separates into one end housing the dendrites and the other end housing the axon terminals or the end bulbs. And these are typically your sensory neurons. So you got a lot of these guys in your skin that are detecting your cutaneous senses. And that's what these are. These are all examples of unipolar neurons. They're all sensory. We've seen or heard of a few of these when we studied skin. This one here in panel A is a Meissner corpuscle for touch. This one here is a lamellated or Pacinian corpuscle for pressure. These were those Merkel tactile structures for tactile senses. And then this guy is a nociceptor. Nociceptors are for pain. So these guys are found, all over the body because you can sense pain internally and externally. And pain neurons are stimulated by a number of things. Extreme temperatures, chemicals like inflammatory chemicals will cause pain. So these guys are your body's way to monitor whether there's a problem that might be occurring. And therefore we can perceive that as pain. Next video we're going to start our more physiology of neuron discussion. where we're going to talk more about these ion channels and how they are involved in your neuron function.